Wild Health is about optimizing you. We use genomics, blood work, biometrics, microbiome assessment, many other tests, and a conversation with you to come up with a full health optimization plan. What's the perfect diet, exercise, and supplement plan for you and only you? The Wild Health podcast is about optimizing all of us. Here we cover the cutting-edge science that gives you the base to be able to apply the personalized plan we give you as a patient. To sign up as a patient, go to wildhealth.com. Or if you're a physician or health coach and you want to learn how to do this for your patients, we're happy to help as well. wildhealth.com for all the information on becoming a patient or working with us.
Hey there, welcome back to the Wild Health Podcast. I have a very exciting guest with me here today, Dr. Morgan Levine, who is the bioinformatics advisor of Elysium Health. Elysium Health, obviously, as most of you probably know, makes the compound basis, which is a NAD precursor. We've chosen to partner with Elysium using their epigenetic clock for all of our patients, and decided that it would be great to have Dr. Levine on the podcast talk with us a little bit about epigenetics and this concept of DNA methylation. Dr. Levine is at the Yale School of Medicine, where she's an assistant professor and aging researcher in the Department of Pathology, and a member of the Yale Combined Program in Computational Biology and Informatics, as well as the Yale Center for Research on Aging. So her lab at Yale combines computational and experimental approaches to identify molecular systems that change in normal aging and disease. Dr. Levine served as a postdoctoral fellow in human genetics at UCLA with Dr. Horvath, which some of you may know, created one of the first clocks, the Horvath clock. Maybe not the first, but probably the best known. She worked with Dr. Horvath and then has continued along the realm of epigenetic aging since then. Yeah, this is such an interesting field, and it feels like it's really exploded over the last few years. So like perfect timing, obviously, for you to go through your PhD and do your postdoc in it. And Pete, there's so much general interest in the community with biological age and longevity at this point. So super cool area to be in.
Yeah, I think I was really lucky to kind of land on this topic. I actually started working on this topic during my PhD, and then since then it's kind of exploded, and it's been really exciting to kind of be a part of that.
Well, thank you so much for being on the podcast. I'm really excited to talk to you today and just pick your brain a little bit about biological age and epigenetics, because I think it's something that, I, as an MD, I obviously learned absolutely nothing about this in medical school, and what I've learned about it has been all self-taught at this point, through reading some research articles and blog posts and things like that. I guess I first personally got interested in epigenetic age with that article that came out maybe like 2 years ago that was basically showing use of like some version of the Horvath clock and improving biological age through treatment with certain interventions, like they were using specifically growth hormone and metformin and DHEA. Mm-hmm. So I'm sure you saw that paper. It was pretty popular, even though it was a very small study. But what I want to do today, if we can, is hopefully just introduce the concept of epigenetic age and maybe talk a little bit about your experience with it and the test that you guys are using over at Elysium for measurement of epigenetic and biological age. So I guess if I could just start with the real basics, our audience is educated consumers. Educated patients for the most part, some physicians. So I will do my best not to ask really specific questions, although I'm very interested in this stuff, so it might happen anyway. But just with that in mind, what exactly is an epigenetic age or a biological age?
So first, just starting broadly, biological age, the way you can think about it is, if I say someone has a biological age of 50, what that means is if you looked in the general population, that they have the biological profile of the average 50-year-old overall. And actually, kind of an analogy I like to use in describing what biological age is you can think of a race, like a 10-mile race. And let's say after 30 minutes, maybe the average person will be 3 miles in. But at 30 minutes, you could also have people who are 6 miles in or even 1 or 2 miles in. So even though chronologically everyone's been running for 30 minutes, or if we made that age 30 years, some people would have, if we say where they are as kind of their biological age, some people would be 60, some people would be 30, some people would be 10. There's variation, even though they have all been around for the same chronological time.
And I guess the first question is, how are we measuring that? What does that measurement tool look like to say how far along in the race they are?
Yeah, so I think, right now we're still trying to figure out, the best way to measure it. So people have developed a bunch of different ways to measure it. Some, from the very simplest, people just do count out the number of kind of deficits or diseases the person has, up to kind of more of these molecular measures that I've been working on, which involve using epigenetic data. So specifically what we do is we look at something called DNA methylation. So these are chemical changes to specific nucleotides in your DNA sequence. And the really interesting thing that people have found over the last few decades is that the pattern of these chemical tags changes very systematically with age. So what we can do is we can look across the genome and say that you have a pattern of these chemical tags that are equivalent to someone who is 50 years old or 30 years old. And the exciting thing is we've actually found that kind of the difference in what your actual chronological age is and what we predict your biological age is based on these chemical tags is actually a good indicator of your health risk. So we can take a bunch of people who are, say, chronologically 40 years old, give them all what we call an epigenetic age, so based on the DNA methylation. And what we find is those who are predicted to be epigenetically 50 have much higher risks for mortality, for a bunch of chronic diseases, than people who are predicted to be 30 or 40, even though they're all the same chronological age.
Right. So let's do some definitions here. So just for specifics, chronological age is basically your birth age, right? So I was born in 1979. I am 40 years old. My epigenetic age is derived directly from methylation sites on my DNA, right? Mm-hmm. And that is basically a laboratory test. I spit into a cup or I give a blood sample. Somebody runs that over to a lab and then looks at my DNA and looks at a specific number of sites to see how many methylated sites there are, and then that gives me an epigenetic age. And then would biological age then be sort of a combination somehow of the epigenetic age and other risk factors? Or can you also say epigenetic age equals biological age?
Yeah, so we like to think of, right now we're kind of using your epigenetic age to proxy your biological age. What I would actually argue is that we don't have one biological age. We have multiple biological ages, because you're going to have different systems, different processes changing at different rates over time. So the way I like to think of it is maybe your epigenetic age is one of your biological ages, but you could have other ones. Some people have used things like telomere length, although there are some issues with the way that's measured in these kind of blood or saliva samples generally. But, you could imagine any number of biological ages that kind of make up your whole mosaic, and that would kind of give you your own personal kind of biological profile, which would map onto kind of your future health and wellness predictions.
So let me just ask a couple of questions about the methylation piece, just to make sure I totally understand it, and maybe help elicit it for my listeners. So the methylation is the addition of basically a carbon and a few hydrogen atoms onto the DNA strand, right? And the cells do this to wrap up DNA, to coil it up so that it can fit basically inside the nucleus. And you have to unwrap the DNA to basically make the RNA to build the proteins, and then you have to wrap it back up. So there's this process of unwrapping and rewrapping, with is basically methylation, and why methylation occurs. Is that correct?
Yeah. So the methylation we look at is on a specific nucleotide, so what we call a CpG site. So this is where you have a cytosine, so the C followed by guanine, a G, along a single strand. And so basically the methylation is laid down as part of a bigger kind of regulatory process in in kind of determining which genes are expressed or how your genome is used. So again, to use an analogy, I like to think of this as kind of a recipe in cooking. So let's say you have a kitchen that you have every ingredient you could possibly need to make whatever it is that you want to make. But what the methylation is doing, or other epigenetic modifications, is determining what your recipe is. So it's saying, put in these 5 things, or how much of the different. It's kind of regulating how much of something you're making or what you're using.
Sure. Right. Just like you wouldn't want to make, like, for example, a troponin in a white blood cell if it's a protein that you use in your heart muscle. So you're sort of, you're turning on and off specific portions of the DNA based on which cellular or which cell you're in, basically. Where is that encoded? Where is that recipe? Is that in the DNA somewhere as well? How does the body even know that?
To determine, actually, we don't know how the body knows which, that this is going to be a neuron, and so I'm going to turn off these parts of the genome and make sure these other parts are available. I think that's not quite clear. And the really exciting stuff that's coming out now with reprogramming is that you can, even if you get a cell that is a fibroblast, you can tell it to go back and be a stem cell and then turn it into any number of cells. So there's some kind of memory that's programmed that you can shift from one cell type to another. But right now, I think we don't necessarily know where that's encoded in kind of the genomic memory.
That's so interesting. There could be all this information, all this knowledge that we don't even really understand yet within the cell in terms of how it even decides to become itself. That's so great.
Yep.
Very, very interesting stuff. So, all right. So in terms of an epigenetic standpoint, we're looking at the number, or the specific CBG sites or methylation sites on the DNA strand, and whether it's methylated or not. And that's different between each cell line, right? So you were sort of hinting to this earlier, that you could have different epigenetic ages depending on which cell line you're looking at. Are we typically seeing people are running on red blood cells, or what cell line are we currently looking at when we do one of these epigenetic tests?
So actually, a lot of the epigenetic measures were developed using whole blood. So all the kind of white blood cells go into it. But again, another really exciting thing we're finding is that at least the epigenetic ages don't seem cell type specific. So I can take an epigenetic age measure that we've developed in blood, apply it in brain cells, and it still tracks with age and predicts really interesting aging phenotypes in brain cells. So there seems to be a more universal kind of pattern that's changing with age, regardless of cell type. Now, there are obviously probably some cell types that this doesn't work in, but for the most part, we've tested, 50 or more different cell types, and we find these consistent patterns using the same exact measure for every type. So it doesn't seem to be cell or tissue specific. On the other hand, I will say the functional implications of this epigenetic aging pattern might be more cell or tissue specific, and I think that's something that we're still trying to kind of sort out.
That's interesting. How is this? How has this technology changed, this epigenetic age changed over the years? I mean, I think everybody's familiar with the concept of the Horvath clock, but truly Dr. Horvath has developed multiple clocks, and now, yourself and others are creating more and more clocks. How is it different?
So the first, what we call epigenetic clock, was developed in 2011, and Steve Horvath was a part of that paper. And what they did was they used DNA methylation measures on about 20,000 of these sites across the genome. And the goal was, can they use that to predict someone's chronological age? And then shortly after, another clock, which we call the Hannum clock, came out. And then the following year, the famous Horvath clock was developed. And what these 3 clocks have in common, again, is that they're trying to predict chronological age. So that is the time since birth for an individual. The difference in the clocks that we're developing now, specifically indexed by Elysium, is that we are actually not predicting chronological time, but the relative time between what we expect between your birth and your death. So are you 75% of the way through your lifespan? Are you 50% of the way? So it takes into account both how long you've been alive, but also how long we are predicting that you will live based on a number of kind of clinical chemistry measures. Another thing that is kind of a shift in the field is now we're trying to create what we call bottom-up measures as opposed to these top-down measures. And so what that means is we're looking at very specific biological hallmarks or features and seeing if we can train those rather than trying to capture more of a global measure. So one example is something called cellular senescence. So this is when cells, either due to stress or once they've kind of reached their turnover potential, go into cellular arrest. And this has a number of potential implications for creating kind of this pro-inflammatory profile. So what we've actually been able to do is create a methylation-based measure for the proportion or the number of senescent cells in a sample. We're also looking at things like kind of an immune or inflammation age, or doing more of kind of a metabolic age. So really specific processes as opposed to kind of this global biological age measure.
I like that. And using your analogy of the race, instead of sort of estimating when the race started, you're now trying to estimate when the race is going to end.
Yes.
Which obviously is much more interesting for all of us who who are trying to figure out how to live longer. We're much more interested in when the race is going to end and how we can push that date forward rather than when we started, because we all know when we started. So I guess one of my questions for you is, how are you even getting that data? I mean, I can't imagine that you've got like a giant, subset of patients where you have epigenetic data and like when they die, so you can you can back calculate their death. How are you even getting to that concept of how much longer they have to create your original data set?
Yeah, so we do have some studies where we have, let's say, 3,000 individuals where we have some information on death for a subset of them. But you're right, a lot of these kind of big long-term cohort studies do not have the methylation data from decades ago. So what we've done is taken kind of a two-step kind of patchwork approach in which we look at these big nationally representative studies where we have decades of information on who lived and when, and if people died, when they died. And we can look at kind of the regular laboratory or clinical chemistry measures, so things like cholesterol, glucose, white blood cell counts. And we actually can make an index of those that predicts how kind of an expected life expectancy for an individual. And then the nice thing is we do have data sets that have those markers matched with methylation. So what we can then do is go to those data sets, and now we don't need the mortality information. We just have this kind of proxy for remaining life expectancy, and we can train to try and predict those measures.
Is it kind of like using the real age test? Are you familiar with that? It's like a long survey list of questions that predicts your real age. Yeah.
So it's a little bit different because it's not like a self-reported survey. It's completely objective blood measurements on people, like ones that you would get at a regular physical exam every year.
Interesting. And so the ultimate goal is to basically report how far along you are, as opposed to just reporting the biological age. Because most of these tests at this point just give you a number, like you send in your blood work and it spits back like 35 or something like that, and and you're happy, or it spits back 47 and you're unhappy. So I guess that's part of my question is, what else can we glean from this? Can we start to report more than just a number back to people?
Yeah, so right now, at least with Index, we're not really reporting anything on kind of predicted life expectancy, because I think a lot of people don't want to think of that in terms of kind of time, counting down time to death. So we are still just reporting back a biological age. But again, this does map on to kind of future health and wellness. But I do think going forward, something I'm really interested in is, again, even if I have two people who are chronologically 50 and biologically 52, they don't have the same profile. We're all unique. And so can we get more kind of granularity and figure out which kind of facets of us are aging faster or slower? So am I more of a metabolic ager, or am I more of an immune ager? And once you can kind of put together, I guess, these multiple biological ages and really paint a much clearer picture of an individual, I think that will really point more towards telling people how they can improve. So if I'm an immune ager, I might do something different than if I'm a metabolic ager. And I think the really exciting thing going forward that we hope to kind of get is, can we predict, based on someone's profile, what interventions are going to be the most beneficial for them going forward? And we can actually track people over time and give them advice rather than just kind of the general advice, exercise, get enough sleep, but more personalized advice based on their profile.
Yeah,
I think that's really where the future hopefully will go.
It's like you took the words right out of my mouth, the interventions. It's almost like you've been doing this for years. Because that's obviously the most interesting thing, right? I mean, like other than just having a number or, having this idea of when the Grim Reaper's coming, obviously you want to know what you can do to change that. So I guess part of my questions are, obviously you're using some sort of objective data, like a laboratory analysis or something like that, to get a sense of, potentially what system is aging the most in a patient. But are you also looking at specific CBG sites to get that data as well? Like, do you look at different CBG sites for a metabolic profile versus an immune profile?
Yeah, so actually, that's something we're working on right now is, using the methylation data, can we map on which ones matter for which kind of profiles? So we'll have one measure that would be more of a metabolic measure, all using the same data. So you sit basically in a tube once, and we can get, hopefully in the future, information on your senescence profile, your metabolic profile, your immune profile, and who knows going forward, there's a number of other ones that we kind of have in the works as well. And it'll be different CPG sites will matter for these different profiles, more or less.
And is it as simple as you look at the CBG sites near the immune genes versus the CBG sites near or surrounding the metabolic genes, or is it much more complicated, and is there more mathematics in it than I'm making it sound?
Yeah, so it's a little more complicated, I think, partly because we don't really understand how the methylome functions or what the different CPG sites are actually biologically doing. So we can't just take, a set of immune genes and look at those specific, like, CPG sites in the promoters of those genes, because it actually seems like CPG sites completely separate, like not even near any immune genes, are somehow important for immune aging. So it's really, what we're really trying to do is, can we kind of group CPGs in terms of which processes they seem to map to in a completely unbiased approach. So not thinking about, what genes are next to or anything like that, but just from a kind of data science approach. And then from there, can we generate more of these measures?
Are you or anyone that you're aware of currently doing any studies looking at lifestyle interventions or pharmaceutical interventions and changes to DNA methylation and epigenetic age?
Yeah, so I think that's where the field absolutely needs to go. I have a few collaborators where we're doing lifestyle interventions. So one, they don't have DNA methylation in this population, but we do have the clinical measures that we use that we predicted with the DNA methylation. So we're imagining that it would work also for the methylation. And for that, we're looking at a fasting mimicking diet, and we see significant decreases in that biological age measure after 2 cycles of this diet. But I think in terms of, looking at exercise interventions or pharmaceutical interventions, so I'm going to be involved a little bit with what's called the TAME trial, which is to test the impact of metformin in slowing biological aging. So, I'm very excited to see kind of what comes out of that, even though it's a little bit of a longer term study. But yeah, I think right now it's not enough to just give someone a biological age or an epigenetic age. It really, if you can't do anything about it, there's kind of no point in knowing, essentially.
Exactly. Yeah. But the cool thing, at least for me, at least in this field, because when we're talking about longevity, obviously, outcome data is very challenging to get, and it takes a really, really long time. And science moves much quicker these days than we can tolerate that kind of waiting. This is an opportunity to kind of bypass that and to get a surrogate of longevity from an intervention. And just like you're describing, 2 episodes of a fasting mimicking diet, or like we saw in the previous paper that's been published with the growth hormone, metformin, DHEA. I mean, that was a cycle of just a few weeks, and we're seeing meaningful epigenetic change. So that, at least for me, gives a validity to the fact that we can change our epigenetic age, and we can, change our expected longevity with these interventions. And it's really cool now to have this tool that we can now use to test on a much more regular interval how we're doing. So I guess, one of my questions is, and this is a question I get from my patients all the time: how often should they be checking their epigenetic age based on interventions? I'm sure you can't really answer that question, but you've got to have a better sense than I do about what sort of time frame are we talking here? Is this something that's reasonable to check every few months, or is it more like a yearly thing?
Yeah, I think, right now, again, because there haven't been these really rigorous kind of what we call longitudinal studies where they're tracking people, in these very specific time intervals, it is a little bit hard to say. Right now, what we're recommending is that people check their epigenetic age every 6 months to a year. For fun, I'm actually doing mine right now once a month just to see how stable it is. And then now I've actually found that it's quite stable. And then for the second half of the year, I'm going to try and do some interventions to then see if I can change that a little bit.
Oh, cool.
Yeah, I would say, though, if someone were to do a major intervention, then, you could check it more regularly. But otherwise, I think it's just good to keep track of maybe every 12 months, perhaps every 6 months, if you really want to be rigorous about it.
You gave me an idea. I'm gonna test it after a 5-day fast. I think that would be a great plan. Just do check it and then fast for 5 days and then check it again immediately and see if there's any significant change. That would be super interesting. But I'm curious what your interventions that you're planning for the second half of the year. You don't have to share if you don't want to, but I think it'd be interesting to know.
Oh no, it's totally fine. Yeah, I'm going to do the fasting mimicking diet, which I've done before, but not in conjunction with this, where, again, that's like a 5-day, not pure fast, but very low calorie fast.
Right.
And then another one is I'm just going to try and up my exercise kind of commitment 1 month, and then see if that has any impact.
Mm-hmm. Would Elysium let you do basis?
Yeah, so the issue is I'm already on. I would probably have to
stop it and then get back on it, right?
Yeah, exactly.
Right. Yeah. No, that would be another interesting one. Obviously, there's a lot of interest in the NAD precursors in the realm of longevity, along with metformin and growth hormone and everything else under the sun these days. But it'll be great to get some data, even if it's, and one thing with this test being fairly affordable is that it does offer itself to these N of 1 studies, because in order to get this data, it's going to take probably large population-based studies where you get, lots of different patients doing the intervention. In reality, maybe what makes the most sense is sort of identifying the areas of concern in each patient based on their serum biomarkers. Say, one patient might be more metabolically deranged, whereas another one has more cardiovascular issues, and then directing your interventions that way, and then monitoring the effect of the interventions through the epigenetic age. Lots to think about here. We've been working on these sort of generalized protocols with our patients for longevity and implementing the epigenetic age tests on them, and that's been really insightful. And people do have different responses to a generic protocol, and I think obviously you're right. It's dependent on what their cause of their increased epigenetic age is, or where their issues are, whether it's metabolic or inflammatory, or cardiovascular, neurocognitive. So this is really interesting. I'm really excited for this field to continue to grow, and I can't wait to see what else it brings. How far away do you think we are from more data from these reports rather than just a number? Is this something that we're going to see in the next year? Are we talking years away? Is it months away?
I mean, I would anticipate, I don't know exactly when Elysium would be planning to put out the new measures, but within the next year, hopefully by the end of 2020, that they'll be adding additional measures that you'll get with your biological age. So yeah, right now we're just making sure that everything's really robust. So actually, I didn't mention this. One thing that we had to work really hard on to actually make this work at the N of 1 level is the reliability of these measures. So unfortunately, we actually found when we went back, when we started out, was that the epigenetic clocks are actually very unreliable at the N of 1 level. So if you implemented the original Horvath clock, or even some of our earlier clocks, if I took one blood sample from you, split it and ran it 2 times, I could get an epigenetic age with a 5 to 7 year difference. And
wow,
that almost derailed the entire thing for us. But luckily, we spent 6 months to a year working on kind of statistical methods to deal with that, and now we get it. So everyone that we test twice, we're getting within 0 to less than half a year difference between their 2 samples. So that's the other difference. I think I don't know exactly what other groups are that are offering epigenetic age measures, but that is something that we really came across and had to overcome. So yeah, I think that every new measure that we put out, we're doing a lot of rigorous analysis just to make sure that it's reliable, that we trust it, and that we want to be publishing all these measures as well to actually provide some empirical validation of them. So making sure that they're informative and predictive of things we think they should be. And then again, getting some indication of whether they might be modifiable. So, again, we don't have a lot of intervention data, but we can look at kind of these large epidemiological studies and say, on average, do people who eat more fruits and vegetables have lower epigenetic age, or people who exercise, or people who have a healthy BMI? And if we do see those trends in our data, we do have some confidence that they probably will be modifiable to intervention.
Yeah, maybe I haven't given enough credit where credit's due. We, at Wild Health, we've moved directly to working only with Elysium regarding our epigenetic age testing. And that's basically because it seemed that your science was as solid as I could find of any of the other commercial products out there. And also a very tight standard deviation, which was a big concern because it's hard to even find published standard deviations from a lot of these other companies. So how do you know how accurate your test is? And if I'm applying it on a patient by patient basis, it's got to be accurate. I mean, just as you said, there can't be a 5-year, variation from sample to sample if I'm trying to figure out whether, the intervention that I implemented 2 months ago is actually functioning or not, right? So exactly. That's the most critical piece of all the pieces. I mean, you could be off by 30 years as long as it's consistent. Like that's what I'm looking for is, did my intervention work or not? So from a clinical standpoint, I really appreciate that. And I really appreciate how open you guys have been regarding the data and just discussion of the product itself. So I'm super impressed with Elysium and the product that you guys have out there right now. I'm really excited for the future and where you guys are going to be taking this in terms of directing intervention to longevity, to gain more longevity and improve epigenetic age. So thank you to you, Morgan, and to Elysium for all the hard work that you guys have done.
Absolutely. Yeah.
But before we end, I want to ask you if you had the opportunity to design just sort of your ideal research study regarding epigenetic age, what would you design? What would it look like? How would you further progress this field?
I mean, I think it comes back to the intervention, the modifiability. I think that's the number one question that we need to kind of figure out is, we can develop all these amazing measures, but unless we know how to kind of shift the dial on them, it doesn't matter. So what I would design would be kind of a longitudinal study that would track people probably in pretty short intervals, again, to get at this question of how often we should be measuring our epigenetic age. And I would have multiple, perhaps intervention arms. So behavioral interventions, perhaps some pharmaceutical interventions, and to really understand what profiles of people respond to which interventions, what parts of our epigenetic age profile do the different interventions change, and also how quickly do they change them. So do you need to be doing, for someone perhaps that switches to a plant-based diet, is it going to take 6 months or a year to really see those benefits, or do you see it after 2 months? These are questions that I think we really need to answer that right now we have no idea about what the correct answer is.
100%. We have zero idea. We're putting it together with biomarkers and subjective findings, and it's nice to have an objective tool to do that. If I could just request one thing, if you also have an arm where you just look at just mindfulness, I think that would be really interesting too, to see what sort of epigenetic change you get from a standard mindfulness practice.
I completely agree. I think kind of the mind-body connection. I think we're so focused on things like diet, exercise, and kind of these pharmaceutical that we forget, things like stress or sleep or, any of these kind of mind-body things have a huge impact on our overall health as well.
Certainly. Well, Dr. Levine, thank you so much for coming on to the podcast today. It's been great talking with you. Really enjoyed this, and maybe we'll have you back on the podcast in the future. We can talk more about some of your advancements in the area.
I'd be happy to. Thanks so much.
Thanks for listening to the Wild Health Podcast. If you found it helpful, give us a 5-star rating and write a review on iTunes. It really helps the podcast, and we greatly appreciate it. If you want to be a client or you want to work with us, go to wildhealth.com. Thanks again.